Hello dear friends, let's move on with our journey towards exploring the various gene regulatory mechanisms and operon or the operon systems. In my last lecture I had explained about the lactose operon as a catabolic operon. In this lecture now we will see yet another complex, beautiful organization of a prokaryotic genome where it is using the gene system to synthesize tryptophan amino acid. So as we saw in the last lecture, any operon system, it is functioning for metabolism. Metabolism, which is made up of two parts, that is catabolic process and the anabolic process. Now lactose as a carbon source which was broken down, that is a large molecule was broken down into smaller molecules, we saw that as lacoperon. Now here this is an anabolic pathway in the sense molecule which is synthesized when in need, like for example amino acid, important amino acid tryptophan. So these small molecules, there are two cases, there can be two instances in the life of a cell where it may have these small molecules, needed molecules, growth factor molecules provided in the environment, provided in the media. In the environment it can have across the cell wall boundary, it may possess these molecules in a ready-made form. If it is available in the ready-made form, then the only thing it has to do is take it inside and build up the large molecules. There can be a second instance for the cell where in the environment these molecules are not provided. If they are not provided, then the cell may have to synthesize it on its own, like the tryptophan operon, which is an anabolic pathway. You can see here tryptophan, it may be provided as a direct nutrient source. Here you can see it may be provided as a direct nutrient source which the cell will take up, and large building blocks like proteins, other macromolecules, cellular structures may be synthesized from these nutrient source, direct nutrient source. As this is an anabolic pathway, there is no question of breaking down but utilizing these molecules to construct a larger cellular component. Now for this, energy is needed. This energy is provided by the carbon source which is continuously broken down, and ATP is released. That ATP is utilized in anabolic pathways. So it is a coupled process, it is all interdependent on one another. And as I say again and again, while understanding these things, you have to bring in your knowledge about genetics, metabolism, enzymology, physiology of the cell, because you see there is a transport of the nutrient, there is breakdown of the nutrient, enzymes are required, so on and so forth. Point here in today's discussion is tryptophan operon is an anabolic pathway. So either it will be directly provided in the cell or it will not be available for the cell. So if it is not available, then it will have to build it on its own. So what are the pathways? First I will just briefly give you a metabolism perspective from where this tryptophan is synthesized. De novo means using a carbon source, it has to synthesize its own tryptophan. Second case is no need for synthesis, means the gene operon system should be off if it is directly made available in the environment. So if tryptophan is available in the media, ready-made availability is there, then naturally its operon should be off. Okay, and if tryptophan is unavailable, means ready-made is not there, then tryptophan should be on. The biosynthetic pathway, anabolic pathway should be put on. There is a third chance here. There is a third probability here which may be encountered by the cell while growing. That is, tryptophan levels can be low. Means it is not enough to shut down a process, operon, but still it is not enough even to initiate a operon system. So what in that case it does? Naturally it has to synthesize its own tryptophan even if there are low levels. So it cannot put it off. So we will see these three stages or three probabilities in a cell, a bacterial cell, in the presence of tryptophan, in the absence of tryptophan, and in low levels of tryptophan. Remember one thing here, this is an anabolic pathway, so the synthesis is important here. So the tryptophan synthesis, I will just give you a brief outlook of how this tryptophan is synthesized by which pathway. The tryptophan pathway, it requires the product of 4 other enzymes or 4 other pathways for completion of tryptophan biosynthesis. You can see the central is chorismate. Chorismate is the common branch point for the production of these metabolites, which include aromatic amino acids, vitamins, vitamin K and vitamin E are produced through this chorismate only. Chorismate is the central ring structure, and this chorismate is produced from the glucose to shikimate pathway. Now this is a separate which you may have studied in metabolism. If not studied, see, it's a interesting pathway where glucose to a aromatic ring structure shikimate is formed, and this becomes the precursor for formation of chorismate. Now at this chorismate is a junction molecule, means from this phenyltyrosine can be produced, coenzyme can be produced, enterobactins can be produced, folates can be produced, certain antibiotics can be produced. But for us in this lecture we are interested in the formation of a amino acid tryptophan. So glucose to shikimate, via shikimate pathway chorismate is formed, and from chorismate to tryptophan 4 enzymes are required. Chorismate is the common branch point for the production of various metabolites, and we will consider it here as a common branch point for the production of tryptophan. Now this shikimate pathway is found in plants, bacteria, fungi, and also few epicomplexan parasites. Epicomplexan parasites are those parasites which possess a very unique organelle called as apicoplast. Okay, it is a type of a plastid, apicoplast, and the best example is plasmodium. This is just an example. Most of the bacteria, they have these pathways, and it is the chorismate molecule or the shikimate pathway is central to the biosynthesis of carboxylic aromatic compounds, chorismate being one of it. So this is just a small perspective of metabolism from for the synthesis of tryptophan, to deal with questions which may pop out in your mind, like from where is chorismate coming, from where is anthranilate coming, from how is tryptophan synthesized from chorismate, what is shikimate, so on and so forth. So I just given you a brief idea which you can explore elsewhere in your studies. So if you see the tryptophan gene order now, the tryptophan operon, you see on the top side tryptophan operon, there are 1, 2, 3, 4 and 5 segments. First is the regulatory gene, second is the promoter gene, third is the operator gene. Just like the lac operon, this comprises of the regulatory gene products. And structural genes, in the lac operon there were 3 regulatory structural genes, sorry. Here there are 1, 2, 3, 4, 5 genes, A, B, C, D and E, and the most noteworthy point here is that all these 5 gene products, which are 5 enzymes, which are 5 proteins, are involved in the production of tryptophan. That is, tryptophan E is synthesized first, tryptophan D next, tryptophan C third, tryptophan B fourth, and tryptophan A fifth enzyme. This is synthetase. So what are the gene products I have tried to explain here in this pathway? This is the tryptophan synthesis pathway. Again, this slide also tells you about a metabolic perspective, metabolism perspective I am showing you for your information, for your knowledge. It is not expected in the paper of genetics that you should draw structures or you should draw this pathway. There is no need. You just remember the products of these genes. I will explain that in the next slides, but here I want to tell you that tryptophan E, D, C, B and A, these are the structural genes. This tryptophan is also regulated just like as the lac operon is regulated with a repressor, and that gene is present here, tryptophan R. So this tryptophan operon is regulated in multiple fashion as compared to the positive and negative regulation which we have seen in earlier lectures. Here this is a very good example of a pathway which is always on. Lac operon was a pathway which is always off, and it is synthesized or it is put on only when lactose is added in the medium. Here, considering the fact that tryptophan may not always be present in the medium, the cell has to constantly synthesize tryptophan. This pathway is always on, and only when tryptophan is available ready-made in the environment, it has to be put off. I'll come to these points. Here just this slide is for you to explain the 5 enzymes which are there. So see here, chorismate or the chorismic acid is the central precursor molecule for the synthesis of end product tryptophan here. One point, those who know feedback inhibition, tryptophan also auto-regulates itself by feedback mechanism also. But here we are trying to understand the genetic control of production of tryptophan. So chorismic acid to anthranilic acid is a reaction which is catalyzed by anthranilate synthetase. This anthranilate synthetase is a product of tryptophan E gene, the first gene tryptophan E gene, and all this is coming from the glucose shikimate pathway. Now first enzyme is anthranilate synthetase. From antranilic acid to next product is N5-phosphoribosyl anthranilate, which is catalyzed by the enzyme phosphoribosyl transferase. Now phosphoribosyl transferase is the product of tryptophan D gene. So tryptophan E synthesizes antranilate synthetase, tryptophan D gene synthesizes phosphoribosyl transferase. From there it is converted to isomerase, PRA isomerase by tryptophan F gene, tryptophan C gene, E, D and now C gene. Tryptophan C gene, there is an intermediate enzyme, but it is a combination effect of the isomerase and synthetase, which converts the N-phosphoribosyl anthranilate to indole 3-glycerol phosphate. So N-phosphoribosyl anthranilate to indole 3-glycerol phosphate. There is an intermediate compound in some organisms. There is species variation of this tryptophan synthesis. Some have an intermediate, so I will delete that part here. So directly N5-phosphoribosyl anthranilate to indole 3-glycerol phosphate by the tryptophan EDC product here. Now see, that is synthase. From synthase to tryptophan A and B, collectively both tryptophan B and tryptophan A both are responsible for the enzyme tryptophan synthetase. Tryptophan B also produces synthetase, tryptophan A also produces synthetase, and this, because there is tryptophan B, it converts indole 3-glycerol phosphate to indole and indole to tryptophan by tryptophan synthetase. So this is the metabolism, this is the metabolic pathway. As a student of genetics, I just want you to remember tryptophan E, anthranilate synthetase, tryptophan D, phosphoribosyl transferase, tryptophan C, synthase, tryptophan A and B, synthetase. Simple. So this is the gene order. Now the tryptophan operon is called as a repressible system, as contrast to the lac operon, which was called as an inducible system. Why this is so? Because lac operon is always off. It is very rare that the cell will encounter lactose. So whenever a lactose comes, that lactose itself induces the operon system for its own breakdown. So we have a system where in lac operon we call it as an inducible operon because it is always off. Now in this anabolic pathway, anabolism is synthesis. The cell constantly requires these molecules for creation of macromolecules. These small molecules will be assembled to create macromolecules like protein. Tryptophan is amino acid. This amino acid will be a part of a larger protein. A protein will be a part of larger cellular organelle, something. So this tryptophan operon is always on, it is always expressed, and it is shut off or it is repressed only when there is presence of tryptophan. Quite interesting and quite contrast with respect to the lac operon, naturally, because that is a catabolic operon, this is an anabolic operon. So tryptophan operon is called as a repressible system. Why it is called as a repressible system? Because this is always on and it is repressed when tryptophan is present in the environment. Okay, so the co-repressor, which is the molecule which acts with the tryptophan repressor, is available when it is made available. When tryptophan is present, it represses the expression of these genes in the operon. So we will see this as a part of regulation. Now, before venturing into the actual gene expression, I want you to understand one thing very clearly. You see this figure here, DNA. So this white part is the regulatory gene and these are the structural genes. Now I have made you very clear what are the products of these genes. Now, having understood these genes, the whole regulation game is just before the expression of tryptophan E gene. Regulation is to control E, D, C, B and G, whether E, D, C, B and A should be put on or not. What are the conditions? I just explained. Now, whatever the explanation of this lecture will follow from here onwards, it will be just before the tryptophan E gene. Even before the tryptophan E arrives, there is regulation. Okay, try to understand. So whatever explanation now I will give from here onwards, it is on the DNA before the tryptophan E gene. Even tryptophan E gene will be transcribed or not, that is the question. E gene will be transcribed only if tryptophan is absent, and E gene will not be transcribed and translated if tryptophan is present. So see here, three figures are there. This is the DNA. On the top, this is DNA. In the middle, this is a coupled transcription and translation, as we have seen it as a process of protein expression, protein synthesis in bacteria. So this DNA, there is this green mRNA being produced, and this green mRNA is being simultaneously translated, and there is a amino acid produced. But remember, again, very important, all these are just before this RNA polymerase enters the tryptophan E gene. So this is only a part of the regulatory DNA I have shown here in the figure. The structural genes will be E, D, C, B, A, which is still not arrived. The ribosome, the RNA polymerase is yet to decide whether it will enter there or not. That is what regulation is all about. If there is need, then it will enter in the tryptophan E region and so on forward. When that will be arised, when tryptophan is not present or present in a low concentration. And if tryptophan is present, then there is no need for this RNA polymerase to enter the E gene, and it has to terminate this mRNA, and that it does by the row independent type of chain termination, which we have seen in gene expression, protein synthesis. The hairpin and loop type of structures, which is also called as the row independent type of transcription termination. This is very important to understand. And here these loop structures, they not only regulate the operon at transcription level, but also at the translation level. As you can see, this mRNA, even before entering the tryptophan E gene, if it forms a terminator hairpin here, that hairpin structure itself is called as a terminator region or a attenuator sequence. Attenuator because it halts some process. So if this is forming near the ribosome in a coupled transcription and translation process, then the blocking of translation takes place. And if this terminator region, terminator hairpin is being formed near the RNA polymerase, then termination of transcription takes place. So this is a very interesting mechanism. You have to remember that tryptophan operon is regulated even before the structural genes are transcribed and translated, even before the first structural gene is reached by the RNA polymerase. There is control. There is control at the transcription level as well as translation level. There is control by formation of attenuator sequences, that is the hairpin and loop structures. Termination of transcription takes place, and even termination of translation of the leader peptide also takes place. So this is a very interesting aspect which we have seen. Now I would like to explain these phenomenon with the help of animation, just like we saw in the lac operon, and then we will come back to the slides for a much more detailed molecular explanation. So come, let's see how this tryptophan, in the presence and the absence of tryptophan, in the low levels of tryptophan, is regulated by the bacterial cell. So before actually going into the details of the regulatory mechanisms, I would like to show you separately how this tryptophan works by regulating at two different levels or two different mechanisms. This is called as the repressor. Just like the lac-operon, there is a production of a repressor, and that repressor controls the tryptophan operon. There is a second mechanism, and which is by attenuation, that is the attenuator sequences. The tryptophan operon in bacteria, it encodes enzymes which are needed for the biosynthesis of amino acid tryptophan. This is the basic. And tryptophan operon is regulated by a repressor and by attenuation. Now attenuator regions are the regions of internal folding of the mRNA. mRNA which is synthesized during transcription, a transcription of a very special region called as the leader region. I'll come to that, but here see this animation and it explains the regulation of tryptophan operon by attenuator sequences. Later on we will see how it is controlled by repressor also, but here attenuator regions are the hairpin and loop structures which are formed during the mRNA synthesis of the coupled transcription and translation of the tryptophan operon. So overall, attenuation in the tryptophan operon can prevent the expression of tryptophan biosynthesis genes. Remember this thing, attenuation structures, that is the stem and loop structures, can prevent the expression of the tryptophan biosynthesis genes when tryptophan levels in the cell they are high. That is when tryptophan is high and when there is a need for turning off of the tryptophan, this thing it functions. So attenuation, it involves certain special structures and sequences of the tryptophan L region. This is a very special region in the regulatory gene. It is called as the tryptophan leader sequences. Later on in the PowerPoint slides, I will give you a very detailed genetic explanation map and the codon sequences of this L region. It is around 130 to 140 base pairs in length, this region, and this is when it is transcribed into a mRNA from the DNA. This tryptophan L region, when it is transcribed into a RNA, it has internal folding in very interesting and special types. There are 4 regions, 1, 2, 3 and 4 regions. 1 and 2 can form an internal complementary hairpin structure. 2 and 3 can form, 3 and 4 can form. Okay, so you can see interactions among the complementary internal sequences of this tryptophan L RNA can form stem and loop structures in the RNA transcript. This is the point. There are 4 regions which are involved here. These are numbered 1, 2, 3 and 4, and these are relative to the 5 end of the, these are numbered relative to the 5 end, means from 5 dash end you start 1, 2, 3, 4 and 5. The 4 region is near the 3. This is the region where the RNA polymerase is bound, means RNA polymerase is bound on to the DNA, a coupled transcription and translation is going on. So 1 is near the 5 dash end and 4 is near the 3 dash end of the gene. So region 2, you can see here, this region 2, it is complementary to both region 1 and 3. Very interesting. Internally, this region 2 can bind either with 1 or can bind with 3. It can form a stem and loop structure, but not with both at the same time naturally. Either it will form with 1, it will not form with 3. And if it forms with 3, it will not form with 1, and 3 will not form with 4 at that time. So very interesting thing this is. So similarly, as you can see, 3 region is complementary to 2 regions, 2 and 4. 2 is complementary to 1 and 3, and 3 is complementary to 2 and 4. 3, if it is forming a stem loop with 4 here, it will be not forming with 2, and if it forms with 2, okay, it forms with 2, then it will not form with 4. So that is, there are 2 to 3 types of structures which can be formed. When region 1 and 2, they form a stem and loop structure, 3 forms a stem and loop with 4, and immediately following 4 there is a uracil rich region which is the attenuator. This sequence, uracil rich region, is called as the attenuator region. Okay, so when the region 2, 3 and 4, as you can see here, when the region 3 and 4 stem is formed, it acts with the attenuator to form a terminator of transcription. In simple terms, if 3 and 4 are formed, termination of transcription will take place. If 2 and 3 are formed and 4 is free, termination of transcription will not take place. So the region 3 and 4, they are very important. If they form a stem and loop, if it acts with the attenuator, that is the uracil rich region, it forms a terminator for transcription at the end of the tryptophan L gene. This is the end of the tryptophan L gene. Okay, so see here, this is what I mean to say. Either the next possibility is when region 2 and 3 they form a stem and loop. Now 1 is free and 4 is free. When region 2 and 3 they form a stem and loop, then the terminator structure does not form. Means this terminator is free, uracil is free. Terminator structures are not formed. So 2 and 3 is not a termination structure. So what it can be called as? It can be called as an anti-termination sequence. 2 and 3 is an anti-termination sequence. 3 and 4 is a terminator sequence. Because formation of 2 and 3 will not dissociate the RNA polymerase, which is not shown in the figure. You can imagine there is RNA polymerase which is attached to the mRNA as well as the DNA, and transcription is going on. Okay, and as I say, this all thing is before the entry of tryptophan E gene. Even before the tryptophan E is encountered, all this action is going on. So we have to decide, the cell has to decide whether to enter into a E or not. Once it enters into E, all the enzymes and all the genes will be transcribed and translated. So here see again, one more time I repeat, when the region 2 and 3 loop is present, the terminator sequence is not formed and transcription will proceed, and tryptophan E and the subsequent genes will be transcribed, translated, and tryptophan biosynthesis genes are put on. Okay, so there are 3 scenarios, 3 possible scenes with respect to attenuation in the tryptophan operon. Which are those? Let us see. 3 possible scenarios. So if transcription is occurring in the absence, okay, occurring, let us say if transcription is occurring alone in the absence of translation, then tryptophan L mRNA will take on most stable configuration. If there is no coupled transcription and translation, see the figure, what is happening? What is happening here? It is the most stable form. What is happened here? RNA polymerase dissociates even before entering into the E gene. Okay, why that has happened? Because 3 and 4 have formed. 3 and 4 have formed. This is the most stable structure, and this always happens. This always happens when tryptophan is present in the medium. Okay, in that form region 1 and 2, 2 and 3 and 4 are formed and transcription is terminated. Now see here what happens. In other scenario, that was the first scenario. In other scenarios which involve the coupled transcription and translation, you can see the question is, now we will start translating this mRNA with the help of a ribosome. Now this mRNA is formed. Of course, there is a ribosome attached here. Whether that will move into tryptophan E or not, that is the question. Now we will start a coupled transcription and translation. We will bind a ribosome here. Ribosome also cannot move forward if there is a stem and loop configuration. So see here what happens. We will add a ribosome. There are now these 2 red lines, if you can see, these are the 2 adjacent tryptophan codons present in the mRNA. There are 2 tryptophan codons. We are talking about synthesis of tryptophan, and very interestingly the mRNA itself contains tryptophan, but this mRNA is a leader mRNA. It is not an essential mRNA, because essential mRNAs will start from tryptophan E onwards. So this is not an essential region, but still there are 2 tryptophan codons present in the leader mRNA. So if the ribosome comes and then it encounters 2 tryptophan, there is no tryptophan, it will stop there. Once if it stops at the 1 region, 2 and 3 will form. Okay, see here. See now the mRNA polymerase, now coupled transcription and translation has started. As the transcription is on, there is a incorporation of ribosome, and it comes to the 1 region, and in one region there are two red lines here which codes for tryptophan amino acid. Now there will be tryptophan, there will not be tryptophan. If there is tryptophan, the protein synthesis should be stopped. If there is no tryptophan, protein synthesis should be on and it should enter the E gene. A very interesting situation. So if there is no tryptophan, then the ribosome will halt here at the one region. At the two adjacent tryptophan codon, the polymerase will halt here. Ribosome, yes, ribosome will halt here. The polymerase is going on and it still has not entered in the tryptophan E gene. So in a coupled transcription and translation process, this ribosome, which has started simultaneously the translation, will halt at these two tryptophan codons, and it waits for the tryptophan molecule. As there is no tryptophan, there is no tRNA molecule, and what happens at the location of the ribosome, this small pause will not give this one for 2. Okay, this will stop this one from getting stem and loop with 2. Thus 2 will form a stem and loop with 3. See here now, as it has stopped at 1, it is not available for 2. 2 and 3 are formed. When this has happened, remember, when there is no tryptophan. Due to no tryptophan, it has stopped here. And 2 and 3 are formed. 4 is free. Transcription proceeds. As transcription proceeds, there is biosynthetic gene production, and this is happening when there is no tryptophan. Okay. Now, if tryptophan levels are high in the cell, if tryptophan is present in the cell, what will happen? What will happen? See here. Now let us see here from here onwards on your screen. If tryptophan levels are high, ribosome will not get to stop at the two regions. See here, see, focus on this two tryptophan codons. Ribosome has started the translation. Now as tryptophan is high in the cell, this will move fast and reach the region 2. This will move fast. See here, it has moved past the tryptophan codon and reached 2. As it reaches the region 2, what happens? See here, as it reaches the region 2, 2 is not available for 3. 3 and 4 are formed. 3 and 4 is the terminator sequence. This is a uracil, repeated uracil molecule. 3 and 4, the terminator, that is the attenuation sequence, is formed, and this leads to the termination of transcription even before the synthesis of tryptophan E. Termination of transcription before the tryptophan biosynthesis genes are reached. This is a very important stage. In this way, attenuation turns off the tryptophan operon when tryptophan levels are high. Okay, now let us see one more animation in a different perspective of the same control of gene expression in tryptophan. So in order for a better understanding, let us see the same things which I have told you right now in another fashion, in some more elaborative and cleaner perspective. So the tryptophan operon, again the same things which I have told, when there is presence of tryptophan and when there is absence of tryptophan. So in the wild type cells, like for example E. coli, which needs to synthesize tryptophan and other amino acids, Monod and his colleagues, they have already shown with the operon system that if tryptophan is present at a sufficient concentration in the growth medium, then bacteria like E. coli will not produce the enzymes needed for the synthesis of additional tryptophan. Repression of the genes which are needed to make the tryptophan synthetase, it allows the bacteria, repression takes place and it allows the bacteria to conserve the energy. Means the bacteria, they never undergo a wasteful synthesis. So if there is sufficient tryptophan in the growth medium, yes or no, that will be the point to decide whether tryptophan will be synthesized or not. Now when there is sufficient tryptophan, no tryptophan synthase is produced. That is the out of the 4-5 pathways which we saw, synthase is considered as enzyme which is responsible for the production at the penultimate step. Okay, so and if it is not present, then no need for the production of that enzyme. The studies, they have revealed that we have seen there are 5 genes and 4 to 5 enzymes which are needed. And Jacob and Monod, they had proposed the repression of the tryptophan operon, which is similar to the induction of lac operon in the presence of lactose. Induction of lac operon in the presence of lactose, and repression of tryptophan when tryptophan is present. The same operon model was proposed. So you can see this tryptophan operon in E. coli, all the components which I already shown, but still again and again I am trying to emphasize for you for a better understanding. The repressor protein here, this repressor protein by itself it cannot bind to the operator, by itself it cannot. It needs induction by tryptophan molecule. So we will call this tryptophan as a molecule which will activate the repressor protein. Okay, so repressor protein by itself it cannot bind to the operator. Tryptophan, if it is present, it binds to the site on the repressor here, and it causes a conformational change here, as you can see on the top, and this allows the repressor to bind to the operator. When the repressor complex is bound to the operator, then transcription of the gene synthesis is blocked. As tryptophan, it participates in the repression, this acts as a co-repressor. As this tryptophan is acting or is participating in the repression of its own synthesis, we will call this tryptophan as a repression, co-repressor. Okay, co-repressor. Now see here, we will see now when there is no tryptophan. When tryptophan is absent, the tryptophan protein here, the tryptophan repressor protein in the yellow color here, needs tryptophan. Okay, in the absence, what happens? See, in its absence it cannot bind. So if it cannot bind, transcription will proceed. Transcription of the genes will proceed, and enzymes needed for biosynthesis of tryptophan will be produced. E, D, C, B, A all will be produced. This is a polycystonic mRNA molecule, and tryptophan operon is on. When tryptophan is absent, repressor is not binding. Tryptophan itself is a co-repressor, not available, no binding, on. Simple. Now you see, if tryptophan is present in the medium, we are talking here about a repressible system. This is a repressible system. If tryptophan is present in the medium, tryptophan binds to the repressor protein, causing an allosteric binding, that is allosteric reaction to occur. The repressor protein will change its site. See here, now it changes its conformational site, which is now ready to bind at the operator. The repressor protein changes the shape in order to bind with the operator of the regulatory region here in the tryptophan operon, and this inhibits the transcription from proceeding. When does this happen? When tryptophan is present in the medium. So there is no transcription, no synthesis of tryptophan genes. All this when tryptophan is present, to avoid a wasteful synthesis. All this is called as the repressible system. Now scientist Dnyanopasak isolated a multi-gene operon, and all this within the leader, means even before the tryptophan E gene is reached, there is a sequence called as a leader, and whatever the transcription and translation of this leader region, remember, transcription as well as translation of this leader region, which is not important. Important are E, A, B, C, D, E genes. Whatever happens with this leader, even if a polypeptide is synthesized, not synthesized, it is leader polypeptides are of no use. They will be degraded and subjected to the amino acid pool. Okay, but this plays a very important role in the control of the transcription and translation, whether it will enter into the E gene or not, E and subsequent genes or not. So Dnyanopasak, he isolated a multi-gene tryptophan operon and found that long sequence, which he termed as the leader sequence. This leader sequence, to be very precise, it is 162 nucleotides. From here, this leader sequence is 162 nucleotides, and it ends at the beginning of tryptophan E gene. Means 163rd nucleotide will be the tryptophan E gene. Within the leader sequence, there is a regulatory site, this poly U, there is a regulatory site called as the attenuator. Attenuator is found as an integral part of the control of mechanism of this operon. So just what I showed you in the earlier animation, the same is there. Now let's just explore it. Attenuation is very important in the gene control of tryptophan operon, and attenuators are present in the leader sequence before the E gene. Leader contains a DNA sequence which upon transcription gives rise to a mRNA which immediately folds into a hairpin loop, as we have seen. So there are 2 to 3 configurations of this hairpin loop, and in the presence of high concentration of tryptophan, the first 140 bases of this 162, 140 are important. 140 bases of the leader sequences which are transcribed, they act as a mRNA attenuator, means they can form a stem and loop configuration. So if the tryptophan is present or absent, repressor is inactive, does not bind to the promoter, what happens we will see. So this is the mRNA which is a product of only the leader region, which is yet to enter the tryptophan E. Okay, so if tryptophan is present, or if present in low concentration, the repressor is inactive and does not bind to the promoter. In that case, what happens is, as you can see, let us see first the high concentration, then we will go on to the low or no concentration. So in high concentration, there is a DNA sequence which transcribes the leader into a small mRNA, and this mRNA molecule folds back itself into these various forms. What I have shown on this slide is not possible. 1, 2, 3, 4 all forming is not possible, because 1 will form with 2 or 2 with 3. 1 and 2, 3 and 4 are possible, and if 2 and 3 is made, 1 and 4 they are free. Okay, we have seen already this. So if tryptophan is present, what happens we will see now. This is just a theoretical model to show you these are the possible types of combinations, internal combinations which has to occur. So there is a translation, coupled transcription and translation. The coupled process is avoided here for clarity, for you to understand. So ribosome, it moves on to the leader mRNA. Remember, this is only the leader mRNA before the E gene is encountered. So there is a DNA sequence present in the leader which gives rise to this molecule, and as the translation proceeds past the 2 tryptophan codons, here there are 2 tryptophan codons in the one, UGG and UGG. These are the 2 tryptophan codons. As tryptophan concentration is high, it is immediately provided to the ribosome and it moves directly into the second gene. As it encounters the second region, 3 and 4 are free to form a stem and loop configuration, because ribosome has very speedily come into the region of 2. 2 is not available for 3. Hairpin forms 3 and 4, and if 3 and 4 are formed, termination of transcription takes place. Termination because high concentration of tryptophan is there. Okay, that is one thing. Now in low concentration of tryptophan, this is the Dnyanopasak model. In low concentration of tryptophan, what happens we will see. This is also very interesting. As the ribosome moves, it remains here, it gets stalled, it gets halted at the 2 tryptophan codons which are present in the just before entering the one region or in the one region. And as there is low concentration or no concentration, even low concentration will not supply here, because that will be needed by the cell somewhere else for building up of the macromolecules. So this ribosome stops. Once it stops, 2 is available for 3 and 4 is free. If 4 is free, termination is not formed, and this coupled transcription and translation moves into the E gene. As it moves into the E gene, all the A, B, C, D, E enzymes are synthesized, and more tryptophan will be synthesized. It will go on and on till the concentration is high. Once it is high, then again regulation takes place. So when there is low concentration or inadequate concentration of tryptophan, the tRNA which is needed here with tryptophan is unavailable. The ribosome, it halts there during the translation, and this influences the secondary structure of 2 and 3 hairpin loop structure, and this keeps the 4 free, and as 4 is free, what happens? Transcription proceeds and there is biosynthesis. So beautifully how the whole mechanism is controlled, it is explained here in this animation. Okay, so we will move on to discuss more in more detail The same things again with some PowerPoint slides where your concepts will be clarified for the third time when it gets repeated. So I hope you enjoyed and understood the tryptophan operon in the animation. Let's see the same things one final time. What happens in a tryptophan operon, how beautifully it is regulated. So you see, when tryptophan is high in the medium, it is called as a repressible system, because when tryptophan is available, this tryptophan operon is turned off. I have explained this already. I am just going through this as a recap with still slides rather than animation, still slides which will help you to draw some figures. So if tryptophan is present, the repressor is active, the operon is off. Therefore, it is called as a repressible system. When tryptophan is high in the cell, it binds with the repressor protein and it changes the conformation so that it becomes active and binds to the promoter region. Binding of the repressor to the near the promoter, that is the operator, overlaps the promoter. So RNA polymerase, here you can see this white molecule, RNA polymerase cannot bind to the promoter, so transcription is stopped. As tryptophan is already high in the cell, there is no transcription of structural gene and biosynthesis is stopped. Therefore, this is a good example of negative regulation. This is a negatively regulated operon. Tryptophan operon is a negatively regulated operon. Lactose was positively regulated. Why negative regulation? Because tryptophan is already there and no transcription of structural gene is required for the biosynthesis. There is no transcription. So negative regulation. Okay. Now if tryptophan is unavailable, means the tryptophan is needed. So a natural anabolic pathway is on. Repressor gene, that is the in the regulatory, you can see the TRPR gene here. See the tryptophan R gene product, which is a repressor, which is originally inactive. In the absence of tryptophan, the transcription of structural gene takes place because this is inactive, cannot bind here, and these 5 enzymes are synthesized. So at low level of tryptophan, the attenuation phenomenon takes place, as we saw in the animation. So here I will focus only this region, L and A region. Okay. If you see the whole operon, tryptophan operon, this is the structure of tryptophan operon. A tryptophan repressor is there. There is a regulatory region. This regulatory region which is there, this regulatory region of the tryptophan operon consists of tryptophan E, D, C, B and A genes. This tryptophan R, which is forming a repressor, is controlling. So that is the repressible system. Now we will see the attenuable system or the attenuation system, which functions only with the leader region. And this leader region is just before the encounter of tryptophan E gene. Even before E gene is encountered, it is regulated by attenuation. So we will focus only on these genes. So at the beginning of the transcribed genes of the tryptophan operon, there is a sequence of at least, I have said, 162 nucleotides, out of which 130 to 40 are important. So these 130 to 40 nucleotides of this leader, they are termed as the leader sequences or the leader mRNA or the leader transcript, which is very important in attenuation. So this is just before the start of the tryptophan E gene. So part of this leader transcript, it encodes for a short polypeptide of 14 amino acids. That is 1, 2, 3 and 4 region which are there. This is all made up of only 14 amino acids. And out of these 14 amino acids, there are 2 amino acids which are adjacent to each other and they are of tryptophan. So see here, we will see this. This is a coupled transcription and translation process which is regulating the synthesis of tryptophan, whether it is needed or not. The translation starts even before the transcription completes, that we know very well. In this attenuation mechanism, the rate of translation determines whether transcription can continue or whether that will terminate. Therefore, this attenuation mechanism, remember, is only found in bacteria. It is an exclusive characteristic feature of the prokaryotes. It is not found in eukaryotes. So this is the tryptophan operon, and as I have highlighted here, the tryptophan leader sequence, which is very important, this 162 base pairs or the 14 amino acid sequence encountered before the tryptophan E gene is called as a tryptophan leader gene. Leader sequences, they play a very important role in attenuation. Leader sequence contains such a nucleotide sequence which mRNA, when it is transcribed into 4 regions, 1, 2, 3 and 4, which can internally fold, and once they form an internal looping. So very perfect nucleotide sequence. If you want, you can have this sequence. This is start from A. From here you will find that this A is the 162nd base, out of which 139 to 40 base. Here these are the attenuator sequences and the termination sequences, and till 140 these are the leader mRNA base sequence. And after 162 or at the 162 there is a tryptophan E, that is the first structural gene starts. So before that, this is the, we call this as a mRNA map, a genetic map sequence of the leader mRNA. So here interesting, as you can see here, in the leader polypeptide you will have 2 tryptophan codons, UGG and UGG, 2 tryptophan codons. So what happens when the RNA, this mRNA will get translated, ribosome will bind here, as you can see AUG, first methionine codon is there. This synthesis is called as leader peptide, which is useless. It is not needed for the structural genes. This is a leader polypeptide which is needed for regulating the structural genes. If it is formed or not formed, it will be degraded and subjected to nucleotide pool. So it is not that important protein molecule. It is important for regulation of transcription. Okay, so ribosome will bind here. First AUG codon, it will go and come. When it reaches the 2 subsequent tryptophan region, here the question is whether it will stop there or it will move on to the second region. If it stops here, then 2 is available for 3. If it doesn't stop and directly comes to 2, then 2 is not available for 3. This itself, whether the ribosome stops or doesn't stop, changes the whole scenario of regulation. It's very interesting. So you can see these are the 4 possible attenuator regions, hairpin loop regions in the mRNA, possible domains, possible 4 domains, possible 4 combinations. Of course, this is a hypothetical structure. 1 and 2 will form and 3 and 4 will form. Not all. Okay, but this is for your understanding. A perfect map with the amino acid is given here. So here 1 and 2 if they are formed, 3 and 4 can form. If 2 and 3 are formed, then 4 is free. We have seen that. So region 3 is complementary to both region 2 and 4. If region 3 and region 4, they pair with each other, they form a stem and loop structure called as attenuator. 3 and 4 are called as attenuators. Okay, why? Because they will allow or not allow, the transcription to proceed in this region. So if pairing occurs between 3 and 2, there is no such attenuator. If pairing is between 2 and 3, there is no attenuator. Only attenuation occurs when there is pairing between 3 and 4. So at the low level of tryptophan, as you can see, or we have already seen in the animation, at low level of tryptophan, this ribosome is stopped because of formation of 2 and 3. Now this is very important. This leader polypeptide is not important. If 2 and 3 are formed, 4 is free and structural genes are initiated. Means ribosome pauses at the tryptophan codon, sequences 2 and 3 stem and loop structure is formed, sequence 4 is free, it prevents the attenuation of 3 and 4, and transcription is on. When does this happen? On. When happens? Low levels of tryptophan. So on, off is controlled by the mRNA internal foldings. This is the important point. So low levels of tryptophan, as you can see, again the same things which have been mentioned here in a much more elaborate fashion. These points are for your notes. You can pause this slide here and take down the notes. The same things are being mentioned again and again for you for the better understanding. This structure, the figure which is taken, this is taken from Microbial Genetics by David Freifilter, which is a very good book for you to understand these mechanisms. Okay, so the transcript of 4 short sequences which are designated 1 to 4 and its functioning, which I have been repeatedly telling you, is given here. So pause this slide, take down the notes, that is for your understanding. Let's move forward. At the final part of this presentation, translation of leader peptide and transcription of tryptophan operon. This shows that regulation takes place at the level of transcription as well as at the level of translation. So when tryptophan is high, when tryptophan is low, this determines whether that will be regulated by transcription or regulated by translation. So I hope you understood all these mechanisms and these figures. These are for your understanding. When the tryptophan is low or absent in the cell, remember it is a coupled transcription and translation. This is a very good figure for you to understand. When tryptophan is low or absent, translation stops. And you see here at this point, when the second figure, this is just figure A is DNA. Figure B is when the tryptophan is low or absent in the cell. What happens? Tryptophan codon 1 it stops, 2 and 3 is found, 4 is free, transcription continues, and here blocking of the leader polypeptide translation takes place. So even at the translation fashion, it is controlling the things. And if 3 and 4 are formed, that is the attenuator, termination of transcription is taking place. So these are the things which you should understand while studying the tryptophan operon. And tryptophan operon is a very beautiful explanation of anabolic operon. But one thing has to be understood here, that see the figure here, there are various types of terminator sequences, not only as simple as we have seen in the E. coli in today's presentation. Survival and replication of most of the bacteria, they require the ability to synthesize various amino acids including tryptophan. And when tryptophan is not available in the media, it has to regulate its own synthesis. So although the reactions of this tryptophan biosynthesis are identical in all the species, gene organization, it is different from bacteria to bacteria. What you just saw in today's presentation was a whole pathway operon, means a polycistronic mRNA molecule where all the 5 enzymes are present in one cluster at one point. Okay, the regulatory mechanisms of these genes may vary from species to species, and there can be dispersed genes also. Gene organization can be whole operon cluster all in one polycistronic mRNA molecule, or there can be dispersed genes also. It is much more complicated, and there can be complex terminator and anti-terminator structures as shown in this figure here. This is just a random example of some bacteria. I don't want to give you much load on your brains explaining all this, but you see this is also such complex hairpin and loop structures of single mRNA are formed. They are called as anti-terminators and terminators. So it's a beautiful world, a complex world out there. The more you try to explore in deep, the more fascinating evidences of things which we can only wonder about are present in this microbial cell, a cell which is invisible to the naked eye, so small, disappearingly small. It exists, functions, and the more you go and explore inside it, the more it starts to begin. The point where you think I understood, that is the beginning of a next higher level of understanding. So you should explore all these things. In my next lecture, I'll just summarize the lac operon and the tryptophan operon in making you understand the regulation of gene expression, how it takes place at the transcriptional level and how it takes place at the translational level. So we will just see the on and off mechanism significance. Okay, already we have seen that. We have seen that in lac operon, we have seen that in tryptophan operon. But just I will take out some parts of that, and we will the main focus will be regulation of a gene. Taking into consideration the bacterial kingdom as a whole, how does the bacteria regulate the gene? It regulates at transcription level, it regulates at translation level. The basic fundamental part of E. coli is, or the motive of the cell is to avoid a wasteful synthesis, one thing, and to put it on when needed and put it off when not needed. That is regulation of gene expression. And remember once again, gene expression is nothing but protein synthesis. Production of enzyme is the motive behind that. Carrying out the metabolic pathways, anabolic and catabolic pathways, regulating it, not allowing the energy to go waste is the main aim behind gene expression. So till then, goodbye, study hard, ask me questions. There are some links given below. Again, as I said, there is one link in which the tryptophan operon is explained in Hindi. You can go through this, do anything, make your fundamental concepts clear, enjoy this molecular level mechanism which exists only in bacteria, only in E. coli. So till the next lecture, goodbye, see you soon.